So we will move to the next speaker, that is Marcus Kaiser. Marcus is, the background of Marcus is biology and computer science, and he did PhD in neuroscience. He works especially in neuroinformatics. He is from Nottingham University, and so is the second speaker of the day. Marcus, please go ahead.
Yeah, okay, one moment. Okay, can you see the screen? Yeah, okay, that works, excellent. Thanks for the introduction and thanks for the invitation to give a talk over there. I will talk about brain connectivity, and we know that the brain is a hierarchical network, but people only looked at one scale of the hierarchy. They only looked at connectivity between different brain regions. And in this talk, I will show you that connectivity within brain regions can be an important component of predicting cognitive function, but in particular, predicting what happens during brain diseases such as epilepsy. So we want to understand how the connectome is changing, how the set of connections in the brain is changing over time. And we can look at healthy development, identifying different features, the folding pattern of the brain, called stratification, the cortical thickness, so what is the thickness of gray matter in different brain regions, and the connectivity in terms of fiber tracts between regions, but also connectivity within regions. We can then look at the trajectory, at the normal trajectory of healthy brains, but also the trajectory for neurodevelopmental disorders. So if people have depression, schizophrenia, or epilepsy, how does network development change for those diseases? So we can simulate how fiber tracts are changing over time, and we can then simulate the dynamics in the system. So we can run population models or Koromoto oscillators or other models to look at changes in brain activity in the system. And based on those models and based on data of individual patients, we want to find out what is the best treatment strategy for individuals. And treatment could be non-invasive brain stimulation, let's say with transcranial magnetic stimulation. It could be focused ultrasound. So this is something we're currently trying in our lab. Or it could be deciding which pharmaceutical drug to give to an individual patient. And if we look at the brain, it is a hierarchical network. So you have at the global level different modules, let's say one module for visual processing, another module for auditory processing, you have the frontal limbic system, and you have the sensory motor system. But then you can zoom in and you can identify further modules. So for example, if you zoom into the visual system, you will see that there was a dorsal pathway for the position of objects and a ventral pathway for the identity of objects, like recognizing objects. And we can zoom in even further and we can find a modular organization within brain regions.
Marcus,
yes.
Mark, you didn't change the slides before? It was only once because...
Yeah, it was only one slide, sorry.
Okay.
Yeah. So basically, we can look at the brain network in higher resolution. We did that for the Human Connectome project. And this is showing you an example of one subject here. If we... Okay, just a moment. So basically, we can look at the whole brain. You can see the left hemisphere in the top left corner and the right hemisphere in the bottom right corner. We can then zoom into one hemisphere, and we can zoom in further into individual brain regions. And as you can see in the center, you basically have a modular organization within brain regions. So you have multiple modules within a region. And those modules might have different functions. If you think about the motor system, there might be one module for arm movement and another module for leg movement. And this is something that we're currently testing for healthy subjects, but we also wonder whether this modular organization within brain regions is informative for brain diseases. And for this, we looked at epilepsy, but before we looked at epilepsy, we looked at the connectivity within and between regions. So the question is this local connectivity an artifact of the processing method? We looked at two regions here, the left superior frontal cortex and the left superior parietal cortex. And you can see across the diagonal the modules that we detected in each of those two brain regions. And over here, you can see the connectivity between those two brain regions. And if there would be an artifact, you would expect that there was some kind of random connectivity between the two regions. But this is not what we find. If we zoom in here, we find that a module in one brain region is connected to a module in another brain region. So there was a very distinct organization of connectivity between those modules in different regions. There was also a distinct spatial organization in that each module within a brain region, so here the left superior frontal region, has the same spatial position. So basically each module more or less corresponds to a patch of cortical tissue on the surface of the brain region. We then moved on to see whether those organizations within brain regions are indicative of brain disorders. And for this, we looked at temporal lobe epilepsy. So we took data from patients and healthy controls. We parcellated the cortical surface into 50,000 nodes and 50,000 triangles, and we matched that with diffusion tensor imaging structural connectivity corresponding to fiber tracts. And we then get two different types of connectivity. On the one hand, you have the low resolution network, and this is what you normally see in the literature. So this is connectivity between brain regions. Okay, so in this case, between 68 cortical regions. On the other hand, we looked at high resolution connectivity. So this is the network with 50,000 nodes, and we looked at connectivity within brain regions. So you can see a zoom in into one brain region here, the fusiform gyrus over here. So you see connectivity within that brain region. And we then find that there are differences between patients and controls. On the left side, you see a control subject. On the right side, you see a patient. And we can look at different network properties, the surface area, the FL, the fiber length, clustering coefficient, local efficiency, global efficiency, and so on. And we can then do comparisons between patients and controls. And when we do that, when we do a group-wise comparison between patients and controls, we find changes for several brain areas. So again, this is only looking at connectivity within brain regions. So we find increases and decreases for several network measures in several regions here. We can then look at the organization of both networks. So this is the high resolution network, but we can also look at the low resolution network and which changes are actually different if we look at the network overall. So we don't look at individual brain regions, but we look at all regions at the same time. If we look at the low resolution parcellation, the connectivity between brain regions, we see some difference in surface area and fiber length. But if we look at high resolution, so connectivity within brain regions at the local level, we see more differences appearing in network measures. So there seem to be more visible changes at this local level within brain regions than at the level between brain regions. And those changes within brain regions increase over time. If we look at the duration of epilepsy, so for how many years do patients have epileptic seizures, if we have a shorter duration, we find that there were fewer brain regions showing those local changes within the region. And if we look at the longer duration, more than 20 years, we first of all see that more regions are showing local changes, and indicated by the size of the sphere, those changes are more severe, they're stronger compared to control subjects. So a future question here is, do those local changes appear before global changes? So at the moment, you would, for example, for dementia, you can look at global changes and you can make predictions whether someone will develop dementia in the next 3 to 5 years. So we had an article about that 2 years ago. But basically, if you look at the local changes, can you make predictions even earlier on? So do you see local changes before you see any global changes between regions? So can there be an early biomarker of later disease onset? And this is something that we're currently start to test for schizophrenia. So schizophrenia normally occurs between the age of 15 to 30. So the question is, can we actually predict whether someone will develop schizophrenia later on, several years before the onset, based on those local changes within regions? We also see that the local changes are indicative of the outcome of surgery. So those temporal lobe epilepsy patients normally would get pharmaceutical drugs, but those drugs are not working. So the patients that we got are severe cases where the drugs don't work, and those would be candidates for surgery where parts of the network are removed. Unfortunately, in 1/3 of the cases, surgery is unsuccessful. So you take out relatively large parts of the brain of one hemisphere, but the patient still has epileptic seizures. So we wondered whether we can actually predict whether surgery will be successful in patients or not. And all those predictions are based on the structural connectivity before surgery. Okay, so we just take the DTI scan, the functional structural connectivity between regions, and we check whether we can predict surgery outcome. So if we first look at good outcome and bad outcome cases, you can see a comparison here. So for good outcome cases, you can see that fewer brain regions show local changes within regions. Okay. And for bad outcomes, those local changes are more widespread in the network, and they are also slightly stronger. But the point is they're more widespread. And this could explain why you have a bad outcome, because you take out parts of the network, but there are still enough regions left showing those local changes and then driving seizures later on. Whereas for the good outcome case, you have fewer regions that are involved, so you have a higher likelihood to take out all of them and stop epileptic seizures. So we can then run predictions where we have within region networks, so those are networks within brain regions at the local level, and we have the standard approach of looking at brain connectivity between brain regions. And overall, we find a better performance in terms of predicting surgery outcome if we look at the local data, changes in the network within brain regions. But importantly, we find a higher specificity. And this means that local changes are better in correctly predicting bad outcomes. And this is very important because this can tell us with 90% accuracy that surgery will be unsuccessful. So this is very important to know because there was a risk involved in surgery. And if you can know beforehand that the surgery will not work in patients, it can either lead to the decision not to perform the surgery at all, or it can lead to the decision to try alternative surgery targets. So maybe you want to look at local connectivity and take out different regions or more regions than you originally planned, and then get a good outcome in the end. So this can be very informative for treatment planning. So future questions are local network features better predictors for what regions should be targeted in surgery? And can those local changes also be predictors for what parts of the brain to stimulate with non-invasive brain stimulation in other diseases? So if you think about using magnetic stimulation or focused ultrasound stimulation, and you want to know which region to target, you would normally look at global brain connectivity, but potentially this local connectivity within regions can give you a better estimate what region or what regions to target as an intervention. So in summary, super resolution within region networks are a novel biomarker for assessing surgery outcomes in epilepsy. And the number of regions and the magnitude of the local changes are indicative of disease progression. And we're currently testing whether those can be early biomarkers of later changes. So can they be biomarkers of cognitive decline for healthy aging or for the progression towards dementia? Can they be biomarkers of later onset of schizophrenia? And those results also hold for different parcellation schemes. So I mentioned that we parcellated the brain into 68 regions, but we get a similar result if we use a higher resolution, if we parcellate the brain into 360 nodes at the level of global connectivity, at the level of connections between brain regions. So we still find that the local level gives a better performance in terms of predicting surgery outcome. There was more information about a brain network development in this book here, which describes features of brain networks. It describes algorithms, how we can get hubs, how we can get modules, how we can get cortical columns, how we can get different features of brain networks. And it then describes how the brain is developing for healthy development and healthy aging, how it develops for neurodevelopmental diseases and neurodegenerative diseases such as Parkinson's and Alzheimer. And it also describes how brain networks are changing with learning and changing with brain stimulation. I would finally like to thank my team, so some current members, some alumni, collaborators, especially on this article, Bernd Weber and Markus Butz-Ostendorf. I would like to thank the funders, and if you want to follow this kind of research, you can follow me on Twitter. So thanks for your attention.
Thank you very much for a nice talk. I don't know if there are questions here in the audience or online. Probably not. There is one question there. Got it.
Just curiosity, I guess. In the beginning of your talk, you showed this picture of brain development. Is that connected with the later part? Is more like analyzing the brain, how it is, and maybe starting to see how it changes over time. But I didn't see the simulation of brain development. Is that something you're developing now, or we see more?
Yes. So it's not part of the talk, but we had a paper last year in cerebral cortex where we basically simulate the development of brain networks at the local level. So if you look at connectivity within one brain region, how do you get multiple layers, and how do you get connectivity there? And in that paper, we found that cell death is really crucial for healthy brain development. So basically half of the cells are dying, and you might think that's a huge waste of resources, right? I mean, first the cells are born, and many of the cells are migrating to different layers, and then they all die. And basically most of them die before birth. So they die before learning and before you have any input from the environment. But basically, if you don't have cell death during network development, you don't get the correct layout of the network. So we basically run simulation, and for that paper we use Biodynamo, which is a platform for simulating not only brain network development, but in general biological tissue. So you can simulate cancer growth with Biodynamo, and the benefit is that it's very scalable. So you can simulate the growth of 10 million cells, including genetic circuits for cell division, including physical interaction between cells, and so on. So this is now used by several groups worldwide to model biological tissue.
Are there more questions? No? Okay, so let's thank Markus and also the Jürgen, so the both speakers of the session. And so thanks a lot.
Okay, thanks.